So with horizontal gene transfer in the prokaryotes, we've talked about transformation and conjugation. And now we're going to take a look at transduction. And remember, transduction, we're going to be using our bacteriophage or virus to take DNA from one bacterial cell and then transfer it to a different bacterial cell. So we're going to need to learn about bacteriophages first before we learn about transduction. Bacteriophages come in two different types. There's virulent bacteriophages and also temperate bacteriophages. Virulent means more severe, and virulent bacteriophages do the lytic cycle. Temperate means moderate, so they're going to be nicer to the bacteria at first. So they're going to do the lysogenic cycle first, and then they'll do the lytic cycle later. With a virulent bacteriophage, it will attach to the outside of the bacterial cell and inject its viral DNA inside the bacterial cell. And then once inside, it will do the lytic cycle. So it will take over the cell's machinery and start making new virulent bacteriophages. And then once the viruses are ready to be released, it will lyse the bacterial cell. And that's why it's called the lytic cycle, and that will kill the bacterial cell. And then the new virus will then go find another bacterial cell to infect and then start the lytic cycle all over again. And so that will be the virulent bacteriophages doing the lytic cycle. If you have a temperate bacteriophage, it's going to do the lysogenic cycle first. So it will bind to the bacterial cell, inject its viral DNA inside, and instead of doing the lytic cycle, it's going to go and do this lysogenic cycle first. So instead of making virus, it's going to insert its viral DNA inside the bacterial chromosome. So this viral DNA is called the prophage. So the prophage is the viral DNA. And then the bacterial cell is now called the lysogen. So the lysogen is just the bacterial cell with the viral DNA inside of it. And when the bacterial cell goes to divide, it will just copy the prophage along with the rest of the chromosome, and then that gets passed to the daughter cells. And then if those daughter cells divide, they will then pass the prophage along to their daughter cells. And so the prophage will just stay dormant inside these bacterial cells until it somehow senses that the bacterial cell is not doing well. And then it will realize that it needs to go find a new host cell because that bacterial cell is going to die. So the prophage will cut itself out of the bacterial chromosome, and then it will enter into the lytic cycle and start making new virus. And then when the virus is ready to be released, it will lyse the bacterial cell, killing it, and then go find a new bacterial cell to infect, and then we'll start the lysogenic cycle over again. So now that we've learned how bacteriophages reproduce, we can learn about how transduction works. There's actually two different forms of transduction, generalized and specialized transduction, but I'm not going to ask you the difference between those. So we're just going to talk about how transduction works, and that's all you really need to know. So in our picture here on the right, we've got a bacteria cell that's being infected by a bacteriophage, and we've got down here on the second step, we've got the viral DNA being made, and our bacterial cell is disintegrating here inside. The bacterial DNA is being chopped up, and here we go, making more viruses inside here. And you can see that most of the process here is going okay. We've packaged up the viral DNA into the protein capsid, but there's been a couple of mistakes here where we've had some bacterial DNA packaged into the viral capsid. And so when this bacterial cell lyses at the end of the lytic cycle and we release the bacteriophages, and one of these bacteriophages with the bacterial DNA infects a different bacterial cell, it's going to inject the other bacterial cell's DNA into this new bacterial cell. And so this bacterial cell now says, hey, I've got this random piece of bacterial DNA, and hey, there's some interesting genes there. So I think I'll incorporate that into my chromosome. And so now you've got recombination that's occurred. You've recombined two different pieces of DNA, and now you've got some genetic diversity. So this bacterial cell is now different from what you started with. And so that's what transduction is. So you've got this bacteriophage, this virus, that has picked up a piece of DNA from a different bacterial cell and has now transferred it to a different bacterial cell. And that is transduction. I'm not going to ask you about specialized transduction and how that is different from generalized transduction. So you can read about it, but I'm not going to ask you about it. All I really want you to know is just transduction. You've got this bacteriophage, a virus, picking up DNA from one bacterial cell and transferring it to a different bacterial cell. And that's how you're transferring bacterial DNA from one bacterial cell to another, is via a bacteriophage or a virus. So I'm not going to ask you the difference between generalized and specialized transduction.